Transmissible cancers are rare but fascinating biological phenomena that occur when cancer cells spread from one individual to another. Research into transmissible cancers, such as those seen in Tasmanian devils, devil facial tumor disease, DFTD, dogs, canine transmissible venereal tumor, CTVT, and marine bivalves, like clams and mussels, is uncovering valuable insights that could revolutionize oncology. Here's how this research may contribute to new treatments. 1. Understanding immune evasion mechanisms. Transmissible cancers must evade the immune system of the recipient host to survive and proliferate. Studying how these cancers bypass immune defenses can reveal novel strategies cancer cells use to avoid detection. Transmissible cancers have evolved ingenious strategies to evade immune detection, enabling them to thrive in a new host despite being foreign tissue. These strategies provide insights into the immune evasion mechanisms employed by cancer cells in general. Below are some of the key strategies revealed by studying transmissible cancers. 1. Downregulation of major histocompatible complex, MHC, molecules. MHC molecules present antigens, including tumor antigens, to immune cells for recognition. Transmissible cancers often downregulate or lose expression of MHC molecules to hide from the immune system. Example: in Tasmanian devils with devil facial tumor disease, DFTD, the tumors downregulate MHC class I molecules, preventing recognition by cytotoxic T cells. 2. Production of immunosuppressive molecules. Transmissible cancers can secrete molecules that suppress the host immune system, creating an immunosuppressive microenvironment. Example: some tumors produce cytokines or chemokines, e.g., TGF, Illinois 10, that inhibit immune cell activation or recruitment, effectively turning off the immune response. 3. Mimicking host tissue. To avoid immune detection, transmissible cancer cells can alter their surface markers to resemble the host's normal cells. This immune mimicry reduces the likelihood of the immune system targeting them as foreign. Example: certain transmissible cancers may re-express proteins or glycans that match the host's normal tissue profile, blending in with the surrounding cells. 4. Genetic and epigenetic adaptations. Transmissible cancers adapt genetically and epigenetically to their host's immune environment. This includes mutations or epigenetic changes that silence immune-activating genes or upregulate genes that promote immune evasion. Example: epigenetic silencing of tumor antigens in transmissible cancers prevents their presentation to immune cells. 5. Avoiding natural killer, NK, cell detection. NK cells target cells with abnormal or absent MHC molecules. To evade these cells, transmissible cancers may upregulate inhibitory ligands that bind NK cell receptors, preventing an attack. Example: the tumors might express high levels of HLA-E or other ligands that signal self to NK cells, inhibiting their activity. 6. Immune tolerance through host compatibility. In some cases, transmissible cancers exploit genetic uniformity in their hosts to avoid detection. Example: Tasmanian devils, which have low genetic diversity, are less able to recognize DFTD cells as foreign due to similarities in their immune markers. 7. Antigen shedding. Some transmissible cancers shed tumor antigens into the bloodstream, diverting immune system attention and preventing effective targeting of tumor cells. Example: by shedding surface antigens, the tumors create a decoy effect, distracting immune cells from the actual cancer cells. 8. Localized immune suppression. Transmissible cancers may alter the local tumor microenvironment to suppress immune activity selectively around the tumor site. Example: reduction of antigen-presenting cell activity or recruitment of regulatory T cells, TREGs, that suppress anti-tumor immunity. 9. Absence of danger signals. Many transmissible cancers avoid triggering immune responses by failing to release danger-associated molecular patterns, DAMPs, that would normally alert the immune system to cellular damage or invasion. Example: tumor cells may suppress stress signals or apoptotic markers to prevent detection by dendritic cells. 10. Recruitment of tolerogenic immune cells. Transmissible cancers can recruit immune cells, like myeloid-derived suppressor cells, MDSCs, that actively suppress immune responses and promote tumor growth. Example: these cells inhibit the activation of T cells and NK cells, giving the tumor a survival advantage. By studying these mechanisms, researchers gain valuable insights into how cancer evades the immune system and how to counteract these strategies. This knowledge can inspire novel therapies to retrain the immune system to recognize and attack cancer, such as immunomodulators, immune checkpoint inhibitors, vaccines, or drugs that block specific immune evasion pathways. 2. Insights into genetic stability and evolution. Transmissible cancers evolve independently of the host's genome, allowing researchers to observe how cancer adapts over time. These insights could identify vulnerabilities that emerge during cancer evolution, highlight conserved pathways critical to cancer survival, help predict resistance to treatments in human cancers. 3. Development of targeted therapies. Studies on transmissible cancers often uncover unique markers or pathways that sustain tumor cells in different environments. This could lead to biomarkers for early detection of certain cancers, targeted drugs designed to exploit these pathways. 4. Advancing immunotherapy. The immune system's response to transmissible cancers can offer lessons for improving immunotherapy. For instance, research on how hosts tolerate or reject transmissible cancers can guide personalized immunotherapy strategies. Lessons learned could enhance therapies like CAR T-cell treatments or therapeutic cancer vaccines. 5. Inspiration for cancer eradication programs. The conservation efforts for species affected by transmissible cancers, like Tasmanian devils, have spurred the development of unique intervention methods, such as population-wide vaccination programs or selective breeding for resistance. These approaches could translate into public health strategies for cancer prevention, community-wide vaccination or screening campaigns for high-risk populations. 6. Model systems for studying cancer metastasis. Transmissible cancers behave similarly to metastatic cells, as they invade and proliferate in a foreign host environment. Research in this area can provide insights into the biology of metastasis in human cancers, lead to therapies targeting metastatic spread, a major cause of cancer mortality. 7. Novel drug discovery. The study of transmissible cancers may identify chemical or biological agents that inhibit tumor growth or transmission. This could lead to drugs with unique mechanisms of action, repurposing of existing drugs for use in oncology. Research into transmissible cancers is more than just a study of rare phenomena. It provides a unique lens to explore cancer biology and potential therapies. By unraveling how these cancers spread and persist, scientists can apply these lessons to improve prevention, diagnosis, and treatment strategies in human oncology.